Thank you, Koei, and also thank you, Jeff, for the invitation. It's a great opportunity for us to share some of our work. So, as mentioned from the title, I'm going to talk to you about how in the last couple of years we've been adding chatbots and LLM features to bioinformatics platforms that we've developed. And this really is done very carefully. So in this, domain, there is this concept of retrieval-augmented generation that you probably heard of, and it's called RAG. And the idea is that, those LLMs, they are really amazing. They're giving you human-like responses. They understand your context and what you would like to do when you ask it various questions. And it's almost like talking to a person, but in our field in biology, biomedical research, it's very dangerous to have them function without any restrictions and making sure that they actually give you the truthful information. So what you do with those RAGs, you have the LLM sort of like understand what you want it to do, but you really constrain the answer by using other resources, typically APIs, databases, knowledge resources that ground the answers. And then you can get verifiable, truthful information that is also citable and tractable. So this is the theme of the whole talk. And I'm going to tell you about 5 different tools and platforms that we've been developing in the last 2 years. And then I will tell you about each one of them, what its purpose, what it is, and then how we added those features to those tools. So you also can be very creative in the way you can potentially incorporate those technologies, those LLMs, chatbot type of helpers into those resources to do some exciting things. So those 5 resources are D2H2, which was initially the intention because this is a diabetes focused webinar series. So we're going to talk about diabetes a little bit, and D2H2, which is a diabetes data and hypothesis hub that we were funded to create by NIDDK. I'm going to talk a little bit about the CFDE Workbench, which is related to this common fund data ecosystem that Koei mentioned, and then the tools Harmonizome, Playbook, Workflow Builders, and GeneSetCart, which are some other databases/apps that also have those LLMs integrated. So we'll start with D2H2, and D2H2 is a website that, I'm going to try to do it live. So I'm going to get out. And I'm actually gonna go to the D2H2 landing page. And this D2H2 site, it came up with this idea of like there are a lot of published, mostly transcriptomics data sets in the diabetes community that are not widely reused. So can we make those resources accessible to the community by having some processing and a way to search through them and have them presented in a website that it's easier to understand what was done with reanalysis. So the site has the ability to query single genes against those data sets and some other tools. You can also submit your gene sets, and then there is a way to browse and search through bulk and single cell studies that are uniformly aligned and processed and presented. And we also added those hypotheses and a chatbot that I'm going to demo. So the first thing is this D2H2 automatically generated daily hypotheses. So if I click on that, This is a hypothesis that is generated every day. So you can go through, this is being generated every day since 2023. But let's just stay with the hypothesis of today. And the hypothesis is taking a gene set that was automatically computed from one of those studies, and it queried it against a resource called RamaGene. And I'm going to tell you a little bit about RamaGene in a second, but RamaGene has a collection of almost 1 million gene sets that were extracted from supplementary materials of publications. And then it looks for gene sets that have high overlap between the D2H2 set and the sets in RamaGene and low abstract similarity. That means that those genes are highly similar. There's a lot of common genes between the two sets, but they're completely from different topics. So in this particular case, there is some pancreatic islets that were treated with high sugar and high fat diet. And then they measure gene expression before and after, like regular chow and this high fat, high sugar diet. And then also, once you take that set of 238 downregulated genes and you submit them to RamaGene, you find a study that profiled retina pigment epithelium cells in response to reactive oxygen species. So, and those are 1,000 genes. And there's a bunch of genes that are overlapping between the two sets, and the overlap is highly significant. So now what we do, we give it to ChatGPT to form an explanation of the apparent high gene overlap and low abstract similarity. So it comes up with an abstract or a paragraph that describes first the two studies, and then it suggests that this possibly could be related to the fact that you stress those cells. So with the high fat, high sugar diet, you stress the pancreatic islets, and then when you treat the retina with reactive oxygen species, you also stress those cells. And typically this is called metabolic stress. And the same genes are downregulated due to this particular stress. Even though the one study used proteomics, the other study used the transcriptomics, those genes are for metabolic pathways that are involved. So it's sort of like that, it does something, you restrict it to what it can do, and it gives you something pretty reasonable. So the next feature is this chatbot. And here it comes up. And then one thing that it's nice, you can actually talk to it. So there is a microphone here, and then it also can talk back. But we'll just use the text, and we'll use some of those canned, pre-made examples. And for example, we'll look at how many studies are done in islets in the mouse. So I'm going to click on this button, and then it will go to ChatGPT. It will ask it that question, and then we will do some processing to make sure that it's a valid question and we can actually answer it. So here it found it needs more information. So it wants to know, do you want to know about bulk RNA-seq and microarray studies, or you want single cell, or you want both? So I'm interested in single cell studies. So I'm going to click on single cell RNA-seq. And here I got a table with all of the single cell RNA-seq studies that are in D2H2. So they're all listed here, and I can scroll through them, and I can pick the ones that I'm interested in, and then I can click on this button, and then that will take me to a landing page of the study. So that landing page has first has a PCA, TSNE, and UMAP of the analysis of the single cell study. So you can see the TSNE look pretty good. You have a bunch of different clusters, and those are likely associated with cell types. And then this is the UMAP. And then here there is a cell type identification. So it says that it found a lot of beta cells. And then here you can actually put in a specific gene and you can see how that gene is expressed across the samples. So for example, this particular gene is only expressed in beta cells and alpha cells. And then you can do differential expression analysis here between the clusters and the cell types. Okay, so this is D2H2. So D2H2, it hasn't really taken off, but it was very innovative, and it was the first time that we created a chatbot, and we decided to adapt it to, so I'm going to go down to the next part of the presentation, which is about the workbench. So the CFDE Workbench, it's a major effort that we've been focused on in the last 2 years, which is about building a data portal for the CFDE. And there is a paper about it in bioRxiv. And this is the work of Daniel Clark, Giacomo Marino, and Errol Evangelista. So again, I can do a live demo of the CFDE Workbench. And the CFDE Workbench actually has two major parts. So based on the RFA, they asked us to divide it into an information portal and a data portal. So the information portal has a lot of information about the consortium, and outreach activities, descriptions of the various parts of the consortium publications, as well as the centers that participate in the CFDE. And then the data portal aggregates metadata and process data from across common fund programs. So the idea of the CFDE is really to have way to bring in all of the data that is collected by various common fund programs and bringing it together for a discovery of new knowledge by combining the data from across the programs. And we also implemented a chatbot here. So this is a search engine that allows you to search across the datasets from across the DCCs or the different common fund programs. And because we are focused today on integrating LLMs and creating those RAGs, we have a very similar chatbot also at the CFD chatbot in that corner. Like you saw, there is an icon for it. So this chatbot is now using the OpenAI assistants. And this is after maybe a year. So D2H2, we created our own sort of like a way to translate the chatbot and integrate it with the LLM. But the OpenAI had a way to do it. So we can give the OpenAI assistants either APIs or documents. So the first thing that we've done is like we gave it a document that described the various common fund programs. And you can click on those icons. So those are participating common fund programs that are part of the CFDE. So if I click on UDN, I should get a paragraph that describes a little bit about this program. And I guess we can move back to the slides because sometimes it takes a little bit. All right, so we got the answer. So here the answer is, just a paragraph that describes what is UDN and what they do and sort of like what are the goals of the program in a very conversational way. The other thing that we could do, those chips give you, again, examples of what can be done. And here we asking it about the expression of AKT, or this kinase that is very well known. We want to ask how it is expressed across GTEx tissues. So here is just asking us, are you sure you want AKT? And we say yes. And then it goes to the GTEx portal using the GTEx API. And then we render bar plot that shows the tissues that this particular gene is either highly or lowly expressed. and then we have this plot. And also there is a way to look at it in the Playbook Workflow Builder, which I'm going to talk about in a second, but let's do another example. So this is we want to look at how potentially we can up or down regulate the expression of STAT3. So this goes to the links, SIGCOMM links portal to find information about mimickers or reversers. And again, it's double checking that we really want STAT3. And then we get a plot and a table of compounds that are potentially up or down regulate the expression of this particular gene. So these are the up drugs and these are the down drugs, and then we can hover them. So it's a little interactive, and you can get that plot. And also, again, you could put it, you could view it in the Playbook Workflow Builder. I think there were some questions in the chat, so maybe I'll pause.
Yes. So, do you want to answer right now, like before moving to next section?
Yeah, let's do that.
Okay, so there's first question from Anita. How can you check for completeness of the chatbot answers? I think this question is coming when you are introduced the D2H2. Yeah.
Yeah, so we do have, like, you can try, so it's very restricted. So the interaction is trying to just make a decision of what the user potentially wanted. And then the answers are already like fixed in a way. So so except that hypothesis part I showed, for the other examples, it's just sort of like tell you, okay, which APIs I can use and what is possible to answer. So we make sure that the answer is complete. So that's
so how do you make sure that the answer is complete? Is this something that you do on a regular basis? Do you check it on a regular basis, or
no, we designed it in a way that we provide the answers ourselves. So the chatbot is sort of like telling us what answer should we give, but the answers are already sort of like baked in on our side.
Mhm.
So we have all the answers, and then they are in a template. So sometimes maybe it's a different gene or it's a different data set, but it's all fixed. And then the chatbot sort of like tells us first, is this a valid question? If it has enough information to answer that question. But once we have that information and it's a valid question, we just give the answer, and the answer is always complete because we guarantee that it's complete because it's just a selection from like which template to use and how to answer that question. We write the answer ourselves.
Okay.
Okay, so the second question is from Yazid. So how does the RAG system handle unstructured biological data such as scientific literature or lab notes?
So just hold on so the way it works, you give it the text, and it's very good at summarizing the text. It can rewrite it for you. It can potentially give you some potential questions about it, but it's not yeah, it's very good at taking this unstructured biological data and the scientific literature and potentially summarizing it and extracting the most, I guess, relevant information from it. So we'll see some more examples later on.
Okay, is that answer your question? Yes. So next question is from the Pablo. So is the data stored in knowledge graph or in vector database?
This is for which question and which part?
Probably it's CFDE part.
Yeah, so it is everything for the CFDE part. Like, for example, the answers, the stuff that is on the screen, it's all coming from the Playbook Workflow Builder. So the first part was coming from a document that described the various centers. And then this part is coming from the Playbook Workflow Builder, which is a good segue for the next section. I'm going to talk about the playbook a little bit.
Okay, and then okay, so she also mentioned it also asked about the D2H2, the knowledge graph and the vector database. Yeah.
Yeah, so D2H2 is stored in a relational database. So we do have a lot of work with knowledge graphs and knowledge graph databases, but D2H2 is a relational, completely relational database. It's a Postgres SQL database.
Yeah, and there is another question. How is domain-specific knowledge, for example, genomics, proteomics, encoded or retrieved in the RAG system for accurate chatbot responses?
Yes, we try to use APIs. So first you want to know what is the question about. Is it about proteomics, genomics, transcriptomics, and then what tools can potentially be used to answer those questions. And then we give we compose an API call based on the question, and then we get the answer from the source. For example, here is the CCOM links or GTEx, and then we can, once we get back the results, we can compose a figure, an answer, based on the data that is returned.
I hope that answers your question. And we have another question. So from Jenny, could you explain a bit about the instances when the RAG also hallucinates and what checks can the user do in this case? Also, when you say when you already have the answer, does it mean it is running a background application which is working well all times?
No, it only get invoked when you get the question. So it's an API call, and it's using a bunch of templates. And we really don't want it to hallucinate. So we are trying to make sure that if it does hallucinate, we give, oh, I could not answer the question because I didn't have enough information. So we don't let it hallucinate. We try to minimize that as much as possible. So it's very restricted in what it can do. And I think you're gonna get more appreciation for that with the playbook. So let's do another session of question and answer in a few minutes. But let me try to go to the next section. So maybe some of those questions will be answered as I go along.
Yeah, sure.
All right. So the next part is about the playbook. And before I actually go to the playbook site, I want to tell you first that this was published last month in PLOS Computational Biology. It was an effort that involved many other Common Fund program representatives, but we were the lead of it, and it's really Daniel Clark that did, the big chunk of the work and also came up with the concept, so he should get a lot of credit for creating that system. And the Playbook Workflow Builder is based on the idea that Common Fund programs, they have a lot of APIs, and you can access their data via those APIs. Those APIs are mostly well documented, and potentially they are linked. So if I take some data from GTEx, I can compute some signatures, and then I can submit them to links to get like drugs. And there's more Common Fund programs than are listed here. And then we created this network of APIs in the background, a database of APIs and how they're connected. And that allows us to build this user interface to build workflows, like bioinformatics workflows that start with, I don't know, a data table, a gene set, and then there's various ways to analyze that data. And I'm going to demonstrate this. So this is the user interface. We're going to show you how to create a workflow in a second. And it really based on those components, Playbook components that people develop independently. So there is this software development kit that allow you to build those components and submit them to the system. So those are workflow. Imagine those like Lego pieces for building a workflow. And there are three types of components. There is a prompt, there's an API resolver, and there is a view. And the view actually generates the figures. The API resolver calls the API and format the answers and formats the API call. And then the prompt gets information from the user, for example, submitting a gene name. And then all of those components, they form like a large network. And then you can explore that network and potentially use that network. So let me go to the actual site, the Playbook site. So this is the Playbook Workflow Builder landing page. And we can start with a gene. And we can start with the same gene, AKT1. And then we select it, and then we submit it. And once we submit it, there's a lot of stuff that you can do with that gene. You can explore information about it. You can look at variants. You can look at other genes that are co-expressed with it. And before we looked at the expression of AKT across GTEx tissues. So we're going to do the same thing. So here I clicked on the button. It actually went to Boston, got the answer already and came back. And then this is the expression of that gene across those tissues. And then what I can do, I can click on this plus sign. And then that gives me different things that I can do with that table. And one of them is to create this vertical bar chart that was similar to what we've seen before. So this produces the vertical bar chart. And now we started with a gene, and then we produce this bar chart based on GTEx tissues. We can go into this report mode, and then here we can change the gene. So I can take this other gene that came with AKT, and now it resolves it, and it shows the expression for this other gene that is a similar gene to AKT, and it can be any gene. So for example, STAT3 is one of our favorite genes. So put STAT3 and then press submit, and you can see that it takes it like maybe less than a second, and now you see the expression. So STAT3 is highly expressed in the lung, for example. So here you have some description of the workflow. So every node in the workflow produce some text that describe what happened. And then that text get concatenated in this description. And you can also get it to be reformat with GPT. And the real sort of like RAG related feature that was added to this is that you can do the same thing with Chatbot. So for example, here it says, show me the expression of ACE2 in healthy human tissues from GTEx. And it's very similar to what we just did. And I can do something similar. So I will tell it, create a bar plot from GTEx to about the expression of AKT1 across human tissues. So this takes a little bit of time to run, but the idea is now what it does, it starts searching this network of Playbook components to find the potential path of clicks that we did to the cards to produce a report that matches what we've seen. So you can see that it sort of like was talking to itself. So this is the line that I entered. And then it says, let's start by selecting a gene. And it already answered AKT1. It was able to extract the gene from the sentence. And then it was also figured out that is GTEx. And then it produces the bar plot. And then it produces this report that we've seen before. Actually, it didn't do it. So I did something wrong, but I was able to do it in the slide. So something that I entered here. So I would like to have a vertical chart that shows the expression of AKT across healthy human GTEx tissues, and that produces this report. So in my example here, it did not fully get it, but it did get the part about GTEx and it got the gene right. And then I can potentially create the bar plot using this additional, breadcrumb. And now I have the same type of report. So it's not perfect, but it tries to compose the workflow using the chat. So I think I showed you all of the RAG features of the Playbook Workflow Builder. And we can move on to the next app. I don't know, maybe we should pause because I did see some questions. So there is some 3 things in the chat that we haven't gone over. So maybe we should pause. I think Janet,
Um,
you had a question.
Janet question already answered. Yeah.
Okay, so we don't have any questions. So I'm going to continue.
Actually, there's one from Stephen. So can this be adapted to explore multi-gene phenotypes rather than only single gene ones?
Yes, so definitely it's already there. You can explore with this feature. So if you go here, I don't know if you have any query that you have in mind, but if you should try it. So I would say which genes are most relevant um, to diabetes based on genes. I'm not sure if it's going to work, but uh, it might have the information in it to be able to produce that. And it might take a minute, but I'm recommend you to try it. And one of the things, this system is flexible enough that it potentially we can potentially add parts for it to make it work. And you can also make recommendation to it. So if you want to suggest things that are missing, there is a place here to suggest a visualization method or algorithm and data transformation method. So here you can just submit a request for something that you would like to have us implement.
Okay, so hope that answers Steve's question. And there's a second question from Janet. For a generic query without specification of a database API, how does the chatbot select which API to retrieve the result from?
So it has this sort of like it has to make a decision. So if it doesn't have enough information, it would stall, but it has to make sort of like a decision if it has. So sometimes it makes guesses and it can make mistakes, but those mistakes are still restricted in some path in the network. So if I go to this component graph. This is this network of components. So it can only find things that are somewhere on the network. So it looks for paths based on the question that you give it, and it gives a suggested path based on what you said. So it will try to make the best guess. And in some cases, it may not be correct. But as the system is used more, the hope is that it will eventually become better at answering and finding the right path. And also, if the pieces, the components are missing, then it potentially can also build those components automatically or have someone implement those missing components to make it better. But right now, it's still maybe not as great as it should be or could be.
Okay, thank you. I don't see other question right now in the chat, so we can move to the next section.
Okay. So the next one is about GeneSetCart, which is a little bit like the playbook, but it is more, it's even easier to use. And GeneSetCart is really a place for you to manage your gene sets and keep them all in one place to be able to analyze them and visualize them. And this is, again, the work of Giacomo Moreno and Stephanie Olaia. And the paper was published very recently in April 10th. So 3 weeks ago, a month ago in Giga Science. So let's try to go to GeneSetCart and then I'll do it live. So this is the interface of GeneSetCart and the features that I really want to highlight. So let me first, I have some stuff in my cart that I want going to delete. So this is how you would see it with like 0 gene sets in your cart. And the features that I want to highlight is this CFDE GMT crossing. So what we've done, we created gene sets from those 8 common fund programs. And some of the interesting ones are we have aging signatures that we created from GTEx. So GTEx has a post-mortem transcriptomics of 54 human tissues from different ages and sexes. So what you can do, you can compute a differential expression on looking at young tissue versus old tissues and how those genes are up or down regulated across the age of the patients, the groups of patients. There's also now a very exciting data set from Motopac, and this is from rats that are subjected to a period of about 2 weeks of endurance training. So they have also profiling all of their tissues or a long list of tissues from rats before and after this endurance exercise regimen. So now what we want to do, we can cross those 2 data sets and potentially find gene sets that are common between the effect of exercise and the effect of aging. And the first 2 most significant overlapping sets are from blood of, comparing young adults from 20 to 29 to 70 to 79 and 20 to 29, 60 to 69. And then female rats, after 2 weeks, those genes are going down in the blood. So both sets are in the blood. Those genes are upregulated with aging and downregulated by exercise. So those are potentially genes that are sort of like bad for you because they are increasing aging and downregulated by exercise, which is known to be healthy or improve your health status. So those genes is 35 genes and 26 genes. What we can do, we can click on this GPT hypothesis and see a little bit about why those genes are potentially, what are they doing? So what is common about those genes? So those gene sets, the overlapping set of 35 genes, is submitted to enricher. And then we also submit an abstract that describes the MotoPack data, an abstract that describes the GTEx data. And then we ask ChatGPT to give us an explanation of why there is such an overlap between those sets. And it really finds that it has to do a lot with the complement system and the platelet aggregation and platelet pathways. So it's something about blood clotting and how maybe some triglycerides type of related to cholesterol and thrombosis. So this is the hypothesis that is generated by those two. But also what you can do, you can take those sets and then I can add them to the cart. And now I have two sets in my cart. So if I go up here, I see the sets here in the cart. And then I can click on this combine and I can do a union, an intersection of those sets to get those genes that are common to those sets. So, and I can add those to the cart. I can also do that with the visualize feature. So if I click on visualize, I can select those two sets and then I can click on this Venn diagram feature and I can see that 24 genes are common. And if I click on that, I can also get them here and I can add them to the cart from here. And then what we have is this generate report. So this generate report can take any number of sets. And here I'm gonna just take one set. And then you can select analysis of those sets with those various tools. If I would select two sets, like here, I also get this ability to form hypotheses with GPT-4. And then let's say I just keep it one. And then I can also click on this generate report. So this generate report query all of those apps and generate images and figures from these apps. It takes a little bit of time and it also creates abstract using ChatGPT. So if I go back to the slides, this is what I showed you already. This is what the report, part of the report look like. So these are transcription factors that regulating those 24 genes. And we can see that there's a lot of nuclear receptors that are regulating those 24 genes that are related to platelet aggregation. And this is the abstract that is automatically generated with the LLM. And the abstract here is also looking at the results. So it tells you about the methods, but also about the results. So the last part, the last tool is Harmonizome. Maybe some of you have heard about Harmonizome. It has a lot of users and a lot of citations. It's now this version 3.0 has RAG sort of like enhancements to it. And this is the work of Ido Diamant and Daniel Clark for the most part. And this update to Harmonizome was published in Nucleic Acid Research in the database issue. So it was published in January of this year. So Harmonizome, basically it's sort of like a repository of process data. So to create a Harmonizome, we go to those databases that have omics data and we convert them into gene sets and attribute tables and abstract the data in a way that potentially can be queried and harmonized. And then this is the number, the state of the data. So almost 100,000 associations between 500,000 attributes that connect human and mouse genes from 81 different resources. So Harmonizome 3.0 as a chatbot that is very similar to the chatbot that I showed, but it's really just a way to query Harmonizome. So here you can say, okay, which genes are most relevant to type 1 diabetes? And then it suggests a couple of GWAS resources. And then you have some buttons to click on as your options. And then you get some answer. Here it found 5 genes with high standard values from GWAS catalog. And then you can click on those genes, and in the very near future, you'll have those LLM generated description of each gene. And to create those descriptions, we are doing something very, I think it's pretty clever, and also sort of like restricting it to stay in context. So we give the top cited articles for each gene to the ChatGPT, and then we ask it to summarize those top 50 most cited papers that mention the gene to find information about the gene. And then it does a really good job with citations describing the gene. And it does like a better job than the descriptions that we get from NCBI. So this is a Harmonizome and the stuff that we're doing with that. So let me just summarize, and then we can answer some more questions. So first, I mentioned how it's very important to do very careful use of those LLMs, but they can really enhance bioinformatics platforms. The usage of those enhancements and the adoption by the community is not very high. And this is partially due to distrust, but also you saw that the quality of those things, it's not 100% bulletproof. It doesn't work all the time. And, our implementation of it needs to improve, but the ideas are definitely there. The Playbook Workflow Builder is this idea of having those building blocks. It can be really generalized, and it can be applied to other fields. And it's sort of like creating a brain that can answer a lot of things very accurately by having this crosstalk between the LLM and this network of APIs and tools that it serves. It's not a knowledge graph, even though it looks a little bit like a knowledge graph. It's Daniel calling it knowledge resolution graph. So it has more like those nodes in that graph that you saw are more than just knowledge graph. They're not just entities, they're actually tools and things that draw knowledge from somewhere. And then we have these ideas of potentially automatically generating those nodes. And then the process data that I showed, there's a lot of like this data that was processed uniformly for harmonization can be used to build foundation models. And this is sort of like where we are now. We're trying to build those foundation models from those process data sets. And I don't want to give too much out of that because that's sort of like our most cutting edge, exciting stuff that we are doing now, and it's not published. And then one of the other things that you saw is this triangulation of evidence from multiple sources. And this is really a powerful way to sort hypotheses and follow the most interesting and promising computational discoveries with experiments. So you can really decide very cleverly about what is the next experiment that should be done by crossing those data sets and finding things, unexpected things that are potentially worth pursuing. We're going to have a webinar next in a few weeks about the Playbook Workflow Builder, and I'm just advertising for it. And this will be circulated, so make sure that you guys have the opportunity to join. And now you have a little bit of a taste of what it is. And finally, I would like to, congratulate my team and the people that work on all of those things that I presented. Obviously, they're doing all the work and they deserve all the credit. So, and also we have funding for various sources, including ITCR, and we have some funding from the Cohen Foundation. We are part of CPTAC, the Common Fund Data Ecosystem, obviously. and then we are part of the Links Data Coordination Center. We were a part of IDG, and we also received funding to develop this D2H2 portal that I mentioned. So I'm going to stop. We still have like 7 minutes.
Yeah, so thank you so much for the fantastic presentation. So now is our Q&A session. For all the audience, you are welcome to unmute yourself to join the discussion. If there's too many people, you can raise your hand and I will assign you to let you to unmute. And there were a few questions during your presentation. The first one is from Paulo. This is during your presentation of Playbook, I believe. Yeah, so it's a follow-up question from the last, from the playbook last slide that you show about the network and then something. So Paulo said maybe add a chain of thought rack. I don't know if it's a question or comment, but yeah, that's the Paulo's question.
It sounds like the chain of thought RAG sounds exciting, but I don't really know what it is. And I don't know, maybe you can share what you have in mind or what it is. Maybe it is a term that is already a common that I just don't know about.
Yeah, so there are racks. Can you hear me?
Yes.
Yeah, so it is something like recently ChatGPT and there's LangChain. So LangChain is something that is that has inbuilt chain of thought. So it will list out how the steps that it took to get to your answer. So someone asks, how does if you ask a question, um, how does the AI and the rack system know which API to fetch? So what chain of rack system does is that it will, if the user has a question, it will list out all the APIs that are available to the rack to query, and then it will also list out why it picked a particular API over others to answer the question. And if the RAG and the AI is listing out all the steps it took to arrive to the answer, then it like the user can ask the next question, and it becomes a engaging query system.
Yeah,
I hope that makes sense.
yeah, it makes a lot of sense. I think it's something that we mostly have implemented because there is a way for us to trace, if you remember those breadcrumbs at the top, tell you
Yeah.
how how the workflow was constructed. I don't think we have the reasoning of why it chose this one versus the other, but it's almost there. But it is a really good suggestion. Thank you.
Thank you.
And then another question is during the GeneSetCart, like list out all the APIs it's searched and why it's decided to use specific API to retrieve response.
I just got the answer to that. Thank you.
Oh, okay. Okay, and then our next question is from Anita. How do you connect gene names over species? This is during the GeneSetCart application. Yeah.
Yeah, so there is in GeneSetCart a way to transform the gene sets from one species to another. So you have the options of keeping the species as it is, like with the IDs that you provide, but it also has a feature to convert between species. And this was done using Biomart and some APIs that were implemented in the background. So we do have that feature. And also you can decide that you don't want to have any gene validation. So you can potentially compare drugs and compare other sets that are not matching. But some of the burden of setting up the gene identifier is on the user. So that we don't do too many like ID mapping for the users. But there are, if you have species IDs, we can convert them. You can convert them automatically within GeneSetCart.
Hope that answer your question. Anita, is that good? Let me see. Okay. And I think the next one, oh, okay. So there's a follow-up question from Anita. So what is the base data coming from?
What does it mean, base data? Is that related to the species?
Yeah, I mean I'm just trying to, because there are so many steps in between these questions and answers, right? I'm just trying to kind of ascertain what are the what are the I mean, yes, there's a button, sure. I can see that, right? But what's underneath that button? What is the translation matrix? What are those databases that actually do the translation between the species, and are those appropriate
Yeah.
decisions?
So I actually, since I did not implement it, I don't have the answer in my forefront, but it is in the paper. So the reviewers asked us to add that feature. So now I'm looking at the landing page, and let me just share my screen. So maybe it's there, but I don't remember exactly how it was done on which database we used. But here is the in this landing page, you can select the species, and then I believe that there is a way to convert from one species to another, because I remember change that it was one of the reviewers' requests. But exactly which, I know that we are using mygene.info and we use Biomart, but I'm not sure an ensemble, they're using ensemble IDs, but I could get back to you with the right answer. I believe that it is in the paper and it is implemented somewhere here.
Okay. And there is another question from Stephen. Can you extend this to dark genes?
Which part? So the dark genes are there. So we cover all of the genes. But I think maybe the question about the feature in Harmonizome that needs enough publications to construct a description. So maybe if I can only guess that Stephen is interested in automatically generated description about a gene that doesn't have any publications, and it is possible. And we know how to predict the functions of those dark genes based on their co-occurrence and co-expression. And we are working on it, and it's already exists in somewhat way in another site that I did not present today.
Yeah, I think that might be answer Stephen's question. And if you have follow-up question, just let me know. And Anita have a follow-up comment. It would be good to understand the chain of data custody with RIDs to give credit to all of the source data.
Yes, so I have a good question for Anita for that. The RRIDs were required by GigaScience. And if you read the paper, the RRIDs are all over the paper. So we had to do it. Like, even though we initially did not have it, it's there. So yeah.
Okay. Wow. So there is a comment from Sarita. So it would be very cool to see all the paths that the Playbook Workflow Builder takes in terms of the actual queries made.
Yes, so if you go to the Playbook website, and you don't use the chatbot part, but you just use the regular interface. Let's say if you start with gene, or let's start something else just to make it interesting. If you start with a gene set, this is an input. So you can see that right now it, there is a gene set. And then once I submitted that gene set, then these are all of the possible paths that it can take. So it has to make a decision out of all of these. And for example, here it has kinase enrichment analysis with KIA 3. So if I click on that, it submits it to KIA 3, and then I get the enriched kinases that are associated with my input gene set. And then once I press on this extend, I have another way to, for example, select the top ranked kinases. So if I only want the top 10 kinases, I can click top 10, and then I can do something with these. So, and then I have to press on this extend. So I clicked on top 20, and then it has some things that you could do with that. So that's how you can sort of like explore what it can do. And then the RAG part, the chatbot part, is going through this process, but so it only tries to expand things that it can handle. So I hope I answered part of that question.